Getting clean, pure, high-quality genomic DNA, or gDNA, from our cells is vital to any sort of genetic analysis. Because of their larger chromosomes and nuclear compartmentalization, purifying gDNA from eukaryotes is a little more involved. When we extract and purify gDNA, we can exploit some of the chemical properties of DNA to eliminate contaminants from our extracts. First thing we're going to do is pellet our protoplasts. There will still be a large amount of goopy, undigested capsule in the way, but we're about to take some steps to make sure that that's removed. First, we decant our supernatant and resuspend our protoplasts in lysine solution. This lysine solution acts as a pH buffer and also contains DTAB, dodecyl trimethylammonium bromide, which is a surfactant like SDS that will act as a detergent, helping to burst open our protoplasts and the organella membranes. We're also going to heat our reaction mix up to 68 degrees Celsius for about 20 minutes, which is hot enough to help dissolve those membranes, but not so hot that it'll start to denature the impure gDNA. Once that incubation is up, we're going to start introducing a solvent system to separate our mix into an organic and an aqueous phase. We're going to head to the fume hood and add an equivalent volume of chloroform. Chloroform helps denature proteins and lipids, separating them from the more polar and therefore water-soluble DNA. After centrifugation, the super non-polar carbohydrates from the capsule should gather here at the interphase between the supernatant, the aqueous phase where the DNA is, and the lower organic phase, which should be full of protein. We're going to take up a little bit of that supernatant, being careful not to interrupt the interphase and pick up any contaminants, and layer on a little bit of a different detergent called CTAB, or cetyl trimethylammonium bromide, which is also a detergent, helping us make sure our DNA mix stays clean. CTAB will form a salt with our DNA, helping it precipitate out of solution. We'll also add a little bit of water. After mixing and centrifuging, we can decant our supernatant and just barely see a small, semi-translucent pellet of CTAB and gDNA. Next, we're going to add some high molarity salt solution to dissolve our pellet and separate out the CTAB. After this, we add a couple of volumes of ice-cold ethanol. The clever trick we're exploiting here is that DNA, especially large pieces of DNA the size of, say, a chromosome, is insoluble in pure ethanol. The low temperature also makes sure that we're further limiting that solubility. As we know, solubility is proportional to temperature, which means that we're leaching off the CTAB and centrifuging down a pellet of just salt and DNA. Once we're confident that milky pellet has finished forming, we can aspirate the supernatant and resuspend the pellet in 70% ethanol. This is the perfect solvent system for this step because there's enough water to dissolve the salt out of our pellet and just enough ethanol to ensure that the DNA stays precipitated out of solution. We spin down again, aspirate the supernatant, and allow the pellet to dry by draining it onto a piece of Kim-wipe tissue. To let the last bit of ethanol evaporate, we usually let our pellets bake at 37 degrees Celsius for a few minutes until it's nice and dry. Now that we've finally got our pellet of purified gDNA, we can either leave it dry or resuspend in a low EDTA tris buffer for further use.